and he has received his MBBS degree from Tanjavur Medical College and diploma in orthopedics and NS from the NCR University. So he has been practicing orthopedic surgeon and have treated a lot of patients related to the polytrauma involving fractured bones, soft tissue injuries, spine injuries, osteoarthritis, rheumatoid arthritis, and many chronic painful conditions related to tendinitis. And he now got interested in regenerative therapy, and from the last 5 years he and his team has evolved multiple treatment modalities related to regenerative medicines using platelet rich plasma and stem cells mostly from the bone marrow as well as the adipose tissue derived stromal vascular fractions for tendinitis and arthritis. And today's his topic is convalescent plasma and stem cells for COVID-19 treatment. Over to you, Dr. Ravi.
Thank you, sir. I'm just sharing my screen. So
Okay.
is it visible, sir?
Yeah, it's visible now.
Yeah. Okay. Make it full screen. So good evening, everybody. I'm really happy and very honored to be in this session among the great scholars, scientists from India and UK. So it's a real good opportunity for the doctors and the scientists to interact and come out with many meaningful treatments. I see a lot of research going on in many universities, not only in India, throughout the world, where there is excellent work on biomaterials like in VIT. I was just hearing about the biomaterials, nanomaterials used for cardiac regeneration, skin and bone regeneration. But in clinical practice, when I am practicing as an orthopedic surgeon, I am not getting many of these research products for practice. So what should we do to get these research products into the market at the earliest? So that will require a combined work between the clinicians and the scientists. So as doctors, we are seeing the patients, we are seeing the struggle, we are seeing the gap which is existing in the treatment. At the same time, we are seeing a lot of work being done in the universities all around us, and people are returning from the Western and Eastern countries after finishing their post-graduation and PhD with a lot of experience. And now this is the time to bridge this gap and get the treatment to the patients at the earliest possible. And COVID-19 has thrown a real challenge to us. So this is high time VIT has organized, VIT and the Swansea University has organized an excellent program where we are interacting with the best of the minds in both fields, clinical as well as the scientific. So today I will discuss about three essential biological modalities of treatment in COVID and the innovations which are part of it. So the first one will be on convalescent plasma, which is picking up recently. Second one will be on stem cell therapy, and the last will be on tissue engineering. So the current modalities, as all of us know, for the acute respiratory distress syndrome, which is the main cause which kills the patient, that is because of the hyperinflammatory response damaging the lung, causing an acute injury, resulting in the loss of the gas exchange in the alveoli. So there is till today no effective drug which can, no approved effective drug for ARDS. There are a lot of antivirals, there are a lot of anti-inflammatories, there are a lot of biologicals like interleukin, monoclonal antibodies on interleukin like IL-6. So all these drugs are being tried for COVID, and the results will be out one by one and we are awaiting the results for many of these treatment modalities. Out of these, the biological treatment with the convalescent plasma and the stem cells, again, there are a lot of studies which have been initiated in almost every country which has got this problem in a large scale. So in India, when we are anticipating the stage 3 and the stage 4 of the spread, when a huge number of people get affected and the hospitals are flooded with lakhs of patients, then that time we will definitely be lacking in the numbers of the medicos, numbers of the paramedical team, the medicines, the kits, the ventilators. So that time we need to prepare for that at the earliest. So in our attempt, we are trying to see what can save the lives. Vaccines can save the lives, but they will take a lot of time. Usually vaccines take minimum 1 to 1.5 years to come out with the successful trial results. And then we have to face the vaccine reactions also. Several drugs like hydroxychloroquine, azithromycin have been tried for prophylaxis, and still we are trying that, though there are conflicting reports coming, with some saying that it is useful and some reports saying they are not helping it. And then there are a lot of antivirals which are being tried, and some of the recent antivirals are showing very good results. But still, the convalescent plasma, which has been tried since 1918, when the first flu in the America, which killed millions of people as a pandemic. So from 1918, people have been trying to use the plasma from the recovered patients. So this convalescent plasma has already been tried in several diseases. It has been tried in several countries. So since, as I said, in 1918, it has been tried in other viral infections like MERS. It has been tried in the SARS also. So, and Ebola, recently it has been tried for Ebola also successfully. Now again, after this onset of COVID, a lot of trials, initially it was started in China, and then now throughout the world, in India, USA, UK, Canada, Europe, a lot of countries are trying this convalescent plasma. So in India, our ICMR has given an emergency approval for this, and they have formed a protocol, and they have approved some centers which can do this trial with convalescent plasma for sick patients who are on ventilator due to this COVID. And we are awaiting the results. Already there are a few successful cases coming out from the Institute of Liver and Biliary Sciences in Delhi, then the private hospitals in Delhi, and then other institutes from Lucknow, and even in Chennai, in our state. Already 10 patients have been put on this convalescent plasma, and Madras Medical College, the premier institute, is one of the approved centers for that. So as I said, this has been a very successful mode of treatment for many viral diseases, and we hope till we get a very good drug, till we get a very good vaccine. So this will bridge the gap, and if nothing is available, then we should be able to produce convalescent plasma in large quantities, so that whenever there is a need, when the need escalates, then we should be able to match the need. So there are a lot of thousands of patients who are getting cured, so we will be able to source many of these patients and get their plasma and convert it into convalescent plasma. So there are, again, as I said, there are a lot of studies which are proving that convalescent plasma will be useful. Now, there is a small addition in our approach to convalescent plasma. So that is, we want to lyophilize this convalescent plasma. So lyophilization of convalescent plasma will make it into a powder form, so that it can be stored for more than what it can be stored as a plasma. As a plasma form, you can store for 1 year. Now, with powder form, you can store up to 5 years. Then it is also the ease with which you can transport this powder form from one place to another. So, so this will be our Brahmastra. I used to say, when you want to win a war, you want one good weapon. With one good weapon, you want to finish off the war. So probably this lyophilized convalescent plasma, if you are able to successfully bring it out, convalescent plasma is already there. This additional step of lyophilization has to be tested and has to be trialed with the ICMR approval. And once we are through those things, then we will be having a product which will be a Brahmastra, which can destroy the virus and save a lot of lives. So as I said, this lyophilized convalescent plasma has got a long shelf life, and because it contains the neutralizing antibody, and it can decrease the viral load. And once you decrease the viral load and you prevent the escalating inflammatory response, you can stop the sequential organ damage, whether it is going to be in the lungs or whether it is going to be in the other organs, because we have seen that other organs like kidney, there is a lot of damage to the kidney, there is a lot of damage which is happening to the heart by way of myocarditis, and people are literally dying because of this sudden death after recovery. People recover from COVID, they go back home, but suddenly they die within a few hours. Probably some of those reasons have been found out to be like myocarditis, which can cause sudden death. So we want this convalescent plasma to be available in every part of the country so that there is the response should be rapid. As I said, when the disease can kill people within a short time, our response has to be rapid. So our proposal, our innovation is in how we are going to make this plasma, convalescent plasma, into a lyophilized form. As I shown in this picture, so once you collect the plasma from the patient, we usually collect after 2 weeks when the patient becomes negative. Suppose there is a patient who had this COVID and then he has recovered from the symptoms. We do the testing and prove that he is negative. This has to be confirmed on the second day. So we need 2 tests on 2 days to confirm that he is cured of the COVID. And once he is cured, he is sent back home. So from that day, it is 15 days time when he is eligible to donate his convalescent plasma. So it will have adequate number of antibodies. It may vary from patient to patient. So you would have read recently there was one doctor, pediatrician in the United Kingdom. So he had the maximum number of neutralizing antibodies. So he's a Guinness World Record holder in the amount of antibodies, neutralizing antibodies he was able to produce. So there is a lot of difference in the amount of antibodies, neutralizing antibodies produced by every patient. So we would like to have a titer above which it is very safe to neutralize the antibodies. So a lot of lab work has to be done where we can quantify. After we take this lyophilized plasma, we have to quantify the antibody titer, and then we should arrive at a titer which will be helpful in neutralizing the viral infection. And then we should be able to convert it into a powder form, as I have shown, by the process of lyophilization. This we have already done for not with convalescent plasma, but we have already done in our lab with a routine plasma when patients donate. We get the plasma from the blood bank, and then we have made a prototype which you can see in a bottle in the powder form. So that is the sample which we have made. The advantage is there are no negative effects. There is a faster reduction in the viral load. And as I said, it is easy to transport as a powder in a vial because it can be transported and stored at room temperature. You can store it in the pharmacies. Whereas if you want to show, if you have to store the plasma, you have to store it only in the cold containers in the blood bank. So once it is made as powder form, it can be stored in every big pharmacy, and it will be easily available for the doctors when it is in a dire need. So it will become an off the shelf product. Once we lyophilize it, it becomes an off the shelf product, and it is very cost effective because there is only one step involved in converting this plasma to into a powder form. So it is very cost effective, and it is already approved in the Indian Pharmacopoeia. Already this process of lyophilization is approved in Indian Pharmacopoeia, and so there is not a big regulatory step involved which can delay the time process. But since it is already approved in the Indian Pharmacopoeia, we can straight away go for lyophilization. And it has to be only clinical testing has to be done in a human trial for the safety with the approval of the ICMR and the DCGI. And it can be easily reconstituted. So that is the advantage of this lyophilized convalescent plasma as against a normal convalescent plasma. So this is, we have a special technology where apart from this reduction of viral load, once you take the plasma in the blood bank, they regularly do the HLA, they regularly do the HIV, HBsAg, and other transmissible diseases testing. So here we add one more step where we reduce the viral load using viral filters, which can prevent the transmission of some unknown viral diseases also, because we are not testing for, say, Zika, we are not testing for dengue, we are not testing for, so some of these chikungunya, some of these viral diseases which can also be transmissible. So there is a process of viral load reduction. So out of which one more process which we are in the which we are trying to do will be adding some of the materials like amatoxolone, where and then exposed to UV light and reduce the viral load. So these are some of the steps which will make this product, lyophilized plasma, much safer than the convalescent plasma which we are currently using from the blood bank. What we are using from the blood bank is not tested for many unknown viral diseases, and there is no viral reduction step involved. Whereas when we make it as a lyophilized plasma, we can add this step and make it much more safer. So as I said, this is a summary of the advantages and this comparison between the conventional convalescent plasma and the lyophilized form. As I said, that can be stored only in blood banks, whereas this can be stored in the pharmacies. There cold storage is needed. Here the room temperature is sufficient. Conventional form is not readily available. As I said, you have to only go to a blood bank to get it. You don't have blood banks in small villages. You don't have blood banks in very small towns. Whereas lyophilized form can be made available, readily available in the pharmacies. And even after getting it into the ICU, you will need minimum 30 minutes to reconstitute, to bring it to the normal temperature. Suppose you get the plasma from the blood bank. So you have to wait for another 30 minutes to thaw it and bring it to the room temperature before we can give to the patient. Whereas the patients need, they are rapidly deteriorating, and we cannot lose time even after getting the plasma. Whereas in the lyophilized form, you hardly require 5 to 10 minutes to reconstitute. We have to add the saline, mix it, and then it is available for use. So as I said, there is a lot of cases. There are a lot of cases which have recovered from COVID. We have got now lakhs of people who have recovered from COVID. All we have to do is educate them. We have to educate the general public and insist on the use of this convalescent plasma. You tell them the significance, of how they can save the lives. One person can save the lives of 4 people. If I donate, I will be able, from my plasma, they will be able to treat 4 people, at least 4 people. So it is a huge opportunity for people who have recovered from COVID in the way they can help their own brothers and sisters. So that education, this education of the general public, and encourage them to donate blood, and then the separation of plasma, that plasma separators are available in many blood banks. So that is the no need for buying a new inventory, only the step of lyophilization, and this has to be done in a certified GMP facility. Even that certified GMP facilities are available in every state. For example, I think CMC Vellore, our chairperson, I think their place has got a certified GMP facility. Again, Stanley Medical College has got a certified GMP facility in Tamil Nadu. So similarly, every state in India has got certified GMP facilities where we can do this lyophilization process. And as I said, the stock can be stored in the pharmacies for immediate use. And can we do it now? Yes, we can do it. As I said, thousands, not thousands. Now it is almost lakhs of recovered patients, blood banks, separators, lyophilizers, and facilities are available. And this is the right time. Is it? What is the ideal time to do it? Accelerate this process. It is right now, because now the recovery rate. No, now a lot of people we are seeing, young people getting affected, and many of them recover. Whereas when old people get affected, the recovery rate is going to fall. So when the second wave or third wave of COVID happens, when older people get affected, we will not get many patients who have got a recovery and a good neutralizing antibody titer. And cost also we have worked out and found that it can be made free in the government hospitals, and even in a private hospital with the cost of production, it can be limited to somewhere around rupees 5,000, which is just a notional number. And the last one is about the challenge of mutation. See, we all know that this COVID this virus can mutate, and already we are seeing a lot of mutated forms. So, how are we going to face this challenge of mutation? That we can do by using the recently recovered patients from the local area. Suppose within Tamil Nadu, suppose I collect from the patients within Tamil Nadu and use it within a very short time, then there is a possibility, instead of using long stored plasma which was taken 6 months back or 1 year back, if we insist on using the recently recovered patient's plasma, then the chances of overcoming this mutation problem is also, we will be successful in that. As I said, there is a process of approval, and in whether it is in the US or in India, so we have to do a safety study, and then we can go for a phase 3 approval from phase 1, or they can give the FDA or the DCGI can give us approval for further studies. And age is no bar for the use of this lyophilized convalescent plasma. Again, no sex restrictions, and then pregnancy also. There are a lot of cases in obstetrics and gynecology where we use the fresh frozen plasma. So in postpartum hemorrhage, which is a very, very fatal indication, fatal condition, when the woman loses a lot of blood, and they use more than 20-30 units of fresh frozen plasma for a single person. So using plasma in pregnancy is already being done, and so that is also again not a contraindication. And as I said, other comorbid conditions like diabetes and hypertension and renal diseases are also no restrictions for using the convalescent plasma. So only some of the few quality assurance tests we should do. As I said, antibody titrate. We should know the titer of the antibody which is available, whether it will be able to reduce the viral load considerably and save the patient. And then the screening, as I said, the HIV, HBsAg, and other viral screening. So with that, I conclude the role of convalescent plasma and the innovation which we suggest as the lyophilization of the convalescent plasma to make it a much more successful product. Next, we come to the role of stem cell therapy in COVID-19. So, as I said, this biological war on corona, this COVID, involves stem cells. So, this is our team of mother cell, where we have got, I am an orthopedic surgeon. So, we have got a dental surgeon, we have got obstetrics and gynecologic specialists, an hepatologist, microbiologist, and then pharmacologist, and Dr. Vasan from Bangalore, who is a MCH urology. So this is a multidisciplinary team who are testing the efficacy of the role and the role of stem cells, which are taken from the bone marrow, which are taken from the fat, taken from the dental pulp, and taken from the cord blood. So we have got the lab, which is shown in the slide. We have got a small facility where we can do the in vitro test, where we do culture of the cells and then study the availability of growth factors, what are all the cytokines which are released from the stem cells, and quantify them. And then we try to find out which source is better for which disease, because you cannot use the same source for all diseases. So the challenge is there are a lot of degenerative diseases of the in orthopedics, like osteoarthritis, in neuro like Alzheimer's disease, Parkinson's disease, and there are a lot of fibrotic diseases like cirrhosis liver, then glomerular sclerosis. So every organ in the body, there are conditions for which we do not have good medicines. We cannot cure. We cannot cure Alzheimer's disease. We cannot cure osteoarthritis. But now, with the advent of stem cells, we are seeing an opportunity of curing some of these diseases using these stem cells. So, as a team, as I said, we have been trying to find out whether this cord blood or dental pulp or adipose tissue or bone marrow, how we can separate the stem cells. This particular slide shows how we collect the adipose tissue, rinse it to remove the excess blood, do the enzymatic digestion of the tissue, and separate the stromal vascular fraction, and remove all the adipocytes and use the stromal vascular fraction. This process takes around 3 hours from the time when we take it from the patient. So within 3 hours, we will be able to get the stromal vascular fraction, which we can use for regeneration purposes, whether it is going to regenerate the cartilage or the bone or the muscle or the hair regeneration or any tissue. So again, another route is what we have used is the dental pulp. Here again, I am showing the isolation of the dental pulp stem cells from the tooth. You can, the same process of adding collagenase and then lysing the cells and taking the pulp cell fraction. We have used it for regeneration of the mandible, and in dentistry, there has been a lot of use of the dental pulp stem cells. One of our own colleagues, Dr. Shankar Narayanan, he was a pioneer in isolation of dental pulp stem cells and using the dental pulp stem cells for some of the conditions as first time in the world. So he has used dental pulp stem cells for few indications like submucous fibrosis. He has used it for osteoradionecrosis, and he has used it for osteoclastoma, which is a tumor affecting the mandible, which can destroy a large part of mandible. And finally, the patient has to undergo a disfiguring surgery. So he has reconstructed he has regenerated the mandible using the dental pulp stem cells, along with some of the scaffolds like tricalcium phosphate and hydroxyapatite. So again, this is one process of isolation from the bone marrow, a schematic representation. Here again, I show after we aspirate from the bone marrow, it takes hardly 45 minutes to separate the bone marrow concentrate, which can be again used for bone regeneration or cartilage regeneration. In all these clinical indications which we have used for the past almost 10 years, what we have observed and what others have observed is the role of angiogenesis. So it is angiogenesis which is the basis for most of the stem cell therapy. This we have proved. We have proved angiogenesis after injecting stem cells into various tissues. Wherever we have injected, whether it is into the muscle, whether it is into the uterus, whether it is into the mucosal, submucosal area, I will just show you pictures how we have proved that angiogenesis has happened after injection of stem cells. So here you are seeing an angiogram of a patient who had thromboangiitis obliterans or Buerger's disease. This is a disease caused by tobacco chewing. So people chew pan parak, then that will affect that will cause atherosclerotic damage to the blood vessels. Here you see on the left side of the two legs, he has got a lack of flow of blood to the ankle and leg, lower part of the leg, ankle and foot, where instead of 3 blood vessels, there are only, usually you have 3 blood vessels, left anterior, this is the anterior tibial artery, peroneal artery, and the posterior tibial artery. Whereas here you can see it is only the anterior tibial artery is present. The other 2 are already blocked. And beyond this point, there is no blood flow. So this person came to us with an ulcer in the foot, close to the great toe. And 4 months it had not healed and he had very severe breast pain. So he was treated by vascular surgeon and the vascular surgeon said, beyond this point, it is not possible to open up the blood vessel. So they can open up the blood vessel if the block is higher up. They can do a bypass surgery. But when you go down to lower levels, it is not possible to do bypass surgery. And so this person was suggested, you better go and have your leg removed. You have amputation done because your ulcer is not going to be healed even after years. So this person came to me for amputation, but I did not do amputation. I took his own bone marrow stem cells, injected it into his leg. So we injected along the course of the artery. You've got a surface marking for all the 3 arteries. For all these 3 arteries, we have a surface marking. So along the surface, if I inject 2 ml of this concentrate, wherever there is hypoxia, these stem cells form new blood vessels. So you can see a lot of collaterals. The main blood vessel will not open up. You will not see that the main vessel will not open up, but you can see a lot of collateral blood vessels which will bring enough blood to the foot. And now it is almost 4 years we have not amputated his leg. A person who came for amputation was not amputated and he is still walking with his own leg 4 years down the lane. So this is another case where we have proved the angiogenesis. This person is a diabetic person where you can see here, this is called the popliteal artery. This popliteal artery is blocked here. There is a block here. So they have already done a bypass. So this is a bypass surgery which was done 4 years back. After 4 years, this bypass also got thrombosed. So 4 years he was doing well, but after 4 years, again, his blood flow below the leg has become very, very less. And he also developed a non-healing ulcer for 4 months and he was having severe pain. He could not sleep in the night. Then the vascular surgeon, they tried one more procedure called the profundoplasty, where they can open up the artery here. They can try to open up the artery here and then try to bring the blood down. That also failed. So the vascular surgeon wrote a letter to me saying that, sir, we have tried all the procedures which are possible. They are now not possible in this person. So kindly do an amputation. So I did not do an amputation. I took his own stem cells from the iliac crest. As I showed in the slide, we prepared the bone marrow concentrate and again we injected along the course of the artery. And here you can see the difference. You can see after 6 months, you can see how much of collaterals are formed. And this person also, after 4 years, he had a second dose also after 2 years. So after 4 years, he is still walking with his own limb. His limb has not been amputated. So this is the advantage of using stem cells, that angiogenesis is possible. So this is the last slide to show about the angiogenesis. This is the ultrasound picture of a uterus. This is the uterus of an infertile person for whom the endometrium, usually the endometrium, has to be a minimum of 8 to 9 millimeters, minimum 9 millimeters, so that the implantation can occur and the zygote can form. If the endometrium is not adequately thick, if they are due to any reason, because of age or because of premature ovarian failure, if they are not forming adequate estrogen and other hormones, if the person has got the endometrium which is only 4 mm or 3 mm or 5 mm thick, that is one of the causes for female infertility. There we have utilized, we have injected the stem cells, and we have shown that it can not only increase the thickness of the endometrium to more than 9 millimeter, it can also form new blood vessels. We can show that angiogenesis by way of the imaging. Ultrasound, we have a Doppler ultrasound which can show that there is an increase in the blood flow, which is again a proof of angiogenesis. Sorry, this is the last slide again for angiogenesis. This is the oral submucous fibrosis, which I was telling you. People take Panparag. When they take Panparag, their mouth, the submucosal area, there is a lot of fibrosis. They are not able to open the mouth. It is called trismus. They cannot eat. They cannot open the mouth and eat. Their salivation also becomes dry. And they, for every morsel of food, they have to take one sip of water. This condition will progress to become oral cancer. So this is called a pre-malignant condition. So here, as I said, my colleague, Dr. Shankar Narayanan, so he has used the patient's own autologous bone marrow concentrate and injected into this area of fibrosed area. This is the fibrosed area. You can see these are all a lot of inflammatory, chronic inflammatory cells, and this is the fibrosed area. It is like a ground glass appearance. Usually, the collagen will be like waves. Normal collagen will have a wavy appearance, but here it is all matted, and you can see small blood vessels which are empty blood vessels. This is because the blood vessels from the fibrotic area disappear. That is called vasoregression. Whereas after injection, you can see there is a lot of new blood vessel formation, and all these chronic inflammatory these inflammatory cells are all gone. So the inflammation is relieved, and here you can see the wavy pattern. See here, this is the wavy normal collagen pattern. So what was fibrosed has been replaced by normal collagen. And again, here also you can see some of the angiogenesis which is happening. So this is the point which I wanted to make. Not only this angiogenesis, here is one more proof that fibrosis can be reversed. So this is from AIIMS, Delhi. So in AIIMS, Delhi Center of Excellence for Stem Cell Research in All India Institute of Delhi. So they have shown that ocular surface reconstruction after corneal fibrosis. Suppose some patient has got acid injury or alkali injury. See, here you can see patient who had an acid injury or alkali injury. If they have fibrosis of the cornea, they take the limbal stem cells, and once they keep the limbal stem cells in the affected portion, they are able to reverse the fibrosis, and they can get back vision. They can get back up to 40 to 60% of their vision back. So these are some of the proofs which I wanted to show that angiogenesis and the tissue regeneration, reversal of fibrosis is possible. So how is it important in COVID? So you are going to see a lot of pulmonary fibrosis. People who had this COVID, who had their ARDS, who had their acute lung injury, and then who recovered, they are going to end up with a lot of fibrosis. Also other organs like kidney or myocarditis, which as I said, so they are also going to end up with a lot of fibrosis, and there is a role for this stem cell therapy in reversing this fibrosis. So as I said, again, there is a lot of recent research on stem cells, and Abu Dhabi Stem Cell Center has already tried 75 patients where they took the stem cells from the peripheral blood and they administered them into intranasal, or if the patient is already in the ventilator, they injected intrabronchial into the trachea, into the respiratory tract. So the other companies like Pluristem, which has got more, almost they claim close to 100% improvement in the small study which they have conducted. Athersis of Australia, they have conducted a trial in which they have come up to 83% success, as against just 8% success in patients who had only the regular treatment, whereas patients who had their stem cell treatment had 83% success in recovering from the acute lung injury from whatever small study they have conducted, and they have published those results. And our own STEMPUCEL in India, STEMPUCEL has submitted a proposal for a trial in India, but they already have entered into an agreement with a European group of companies, and they are trying for COVID in Europe as a consortium. They are trying the STEMPUCEL, which is actually a product which they designed for critical limb ischemia. No? As I showed the first slide of angiogenesis, where a patient had tobacco taking, and then his leg lost the blood flow, and he was suggested amputation. So STEMPUCEL has come out with a product for the critical limb ischemia, which they are now trying for the COVID. As I said, this COVID pathology, there are a lot of learning happening throughout the world. So it is not a single disease. It is not affecting a single organ, and it is affecting the other systems in multiple ways. But whatever is the way, the alveolar cells are damaged. So you've got a lung damage, where you've got lung tissue damage, where there are alveolar cells are destroyed to a large extent. There is a lot of edema, gas exchange is not happening, and there are microthrombi which are seen in the blood vessels, which are seen in the arteries, arterioles, capillaries, and venules in the lung tissue. So how are we going to address all these problems of inflammation, loss of tissue, and microthrombi? So now they have started, some of the centers, they have started using antithrombotic agents, and there are centers which are only using the anti-IL-6, the predominant cytokine which is destroying the lung tissue. So we have got drugs. Tocilizumab is a drug which we use to address the issue of cytokine storm. So there are drugs which can act at one point, but they cannot act in regeneration. So if you, after stopping the inflammation, how are you going to help in regeneration? There is loss of tissue. So how are you going to help in regeneration? So that is where the role of stem cells happen. And that is how probably the Abu Dhabi Stem Cell Center has been successful in delivering the stem cells to patients who are on ventilator, who had damaged their alveolar cells. These stem cells probably are able to help in the regeneration. So how do? They do it by altering the host immune system. And then we know how the cycle of inflammation sets in, and then ultimately ends up in a cytokine storm. Though our T cells and B cells are able to produce the antibodies and the cellular immune mechanisms which can counter the viral damage, beyond a point, the body is not able to succeed in stopping the damage, and that is where the patients end up in ICU and ultimately die. So how these stem cells can help in regeneration? There are a lot of proposed mechanisms how these MSCs interact with the host immune cells and release the cytokines. All of us know that these mesenchymal stem cells secrete a lot of cytokines, and they are immunomodulatory in nature, and then they are mitogenic, so they can improve the regeneration of new alveolar cells, and they also have the anti-fibrotic property, where they initially secrete lot of matrix metalloprotease enzymes and remove the damaged portion, and then they secrete the tissue inhibitor of matrix metallase protease enzyme, where they can start the process of relaying the collagen and re-establishing the extracellular tissue area, which is responsible for the gaseous exchange. So the damage and the repair has to involve the epithelium, the alveolar epithelium, the endothelium, as well as the tissue in between the two, which is the respiratory membrane. So there are a lot of registered clinical trials, as I said, currently, which are undergoing phase 1, phase 2 studies in several parts of the world. So we are expecting some of the results. Some of them are out with their preliminary results, and more is yet to come. So the last part of this lecture is going to be on tissue engineering. So this is only a small part where already even in India, there are companies which have come out with lung organoids. So they have come out with small 3D structures of lung. They have made lung tissue. They have engineered lung tissue for the sake of study of drugs. Now the same can be used for treatment also. So these are the two institutions which I found, the University of Pennsylvania, and in our own country, in Bangalore, we have got iStem, which is a company doing lung organoids. So as I said, this is the company iStem, and their product of lung organoid is now being utilized in Europe for study of the new drugs which can be used in COVID. So this is sourced from the induced pluripotent stem cells. So iStem has been able to produce these lung organoids 3D structures from the induced pluri stem cells. So this is one approach where again we can succeed. The same 3D spheroids can be useful in the treatment also. So we are trying to develop some of these organoids and try how we can treat not only COVID, the other damages and other regeneration process using the 3D tissue engineered structures. So this brings me to the end of the lecture, where I just wanted to say about the biological treatments of convalescent plasma stem cells and tissue engineering, addressing the COVID as well as the future challenge. Thank you.
So thank you, Dr. Ravi, for a very nice talk. And we have a lot of questions. I will try to address some of the questions and some of the important questions. One of the questions that one of the participant has asked is, can stem cell help to manage the cytokine storm after the COVID infection? If yes, how?
I'm not sure about the cytokine storm, because that happens very fast. So whether we are able to dose the cytokine storm adequately, that is the question. So definitely the cytokine storm can be addressed by the MSCs, because we know there are a lot of anti-inflammatory interleukins and the cytokines which are being secreted by the mesenchymal stem cells, which can address the problem of cytokine storm. But every patient is different. So at what stage we give these MSCs, whether the dose which we give will be adequate to counter the storm and completely recover. So that will be a challenge, and that needs to be studied in a clinical trial setting in multiple doses. So we have to try it in multiple doses. Nobody knows the exact dose what you have to give for a particular patient. So this needs a lot of studies. So in short, the answer is yes, but we don't know the dose. It needs studies.
Absolutely. So we are trying in some experiments related to the acute radiation syndrome Actually, where we use human water jelly derived MSCs for the treatment in mice model, and we see it's controlling the cytokine storm to a level there where the some of the resistant stem cell itself is able to mount a regenerative process.
Yeah.
So second question we are going to take is, even though the plasma generates continuously for human, by taking 400 ml of plasma from a person will cause any effect on the person's immunity or not?
Actually, when they do the plasma separation, they only remove the plasma. They give back the cells. So in a plasmapheresis machine, so they take the plasma and give back the RBCs, WBCs, and all. So the B cells will be returned, and they will be able to make the antibodies. Since the patient has already recovered, there is no need for the neutralizing antibody at present, but for a second infection, he should be ready. So I don't know the vulnerability. The problem is if he acquires an infection immediately, which is a possibility because we are living in our family. There may be another member who is affected with COVID. He might not have recovered. I might have recovered. He might not have recovered.
He may be asymptomatic here.
Correct. He may be asymptomatic. So again, as I said, so this is a challenge. So that is why I said we should be able to measure the antibody titer and tell at which titer level is we are safe. So we should be able to give the patient as well as retain. Suppose I am able to retain a reasonable titer of antibody which I find is adequate, then I will be saved from the future attack also.
Sure. So the next question is, plasma therapy is used to treat COVID, but are there any chance that the tissue of the patient may reject the treatment?
Come again, sir.
So the plasma therapy is used to treat COVID, but are there any chance that tissues of the patient, means the tissues of the receiving the treatment, may reject the treatment?
No, actually, this plasma we have been using for several years for several diseases. As I said, for obstetrics, we are using 20 to 30 units of plasma for a single patient. So only when the plasma has some retained cells, suppose you are not able to filter the cells completely 100%, then there is a possibility that the cellular component can mount an inflammatory response. And there are accidents which have happened when we give 30 to 40 units. So that is a possibility if there are some cells which are not filtered, if it is not 100% filtered.
And the next question is, what would be the dose of plasma and the frequency at which it has to be administered?
That will be clinically decided by the doctor who is treating the patient. So it may be 1 unit, it may be 2 units with a given in a gap of 1 week. So that will be individualized to the patient and will be it will be decided by the doctor, with the treating doctor.
So the next question is a little more challenging, because does this treatment, the plasma treatment, cause any autoimmune diseases?
Again, as I said, there is no chance for autoimmune disease because of this. Again, the same answer is that if there is a mixture of cells, if the filtration is not proper, then there is a possibility. There are, when we give blood transfusion, we have seen reactions, patient developing immediate reactions and die, or people who get reactions later and autoimmune disease. So that possibility is only if
maybe then
the process.
Maybe then we have to develop a little bit more quality control.
Exactly.
Quality control.
Correct. That's right.
To release the product. The released product has to be very well QC.
Correct.
So the next question comes is, what are the inclusion and exclusion criteria for the candidate who can receive this and an ideal donor?
Is this regarding plasma therapy?
Yeah, plasma therapy.
Okay. So the ICMR itself has given the guidelines for the donor as well as for the recipient. Like age, they should be at least 18 years, and then the lab confirmation. The patient should have had a confirmed disease. Then they should have confirmed negative. Should have become confirmed negative. So there are a lot of these inclusion criteria, the selection criteria for the donors, and similarly for the patients who are going to receive, what is the approval which the institution asked for, whether if they wanted to give this plasma therapy for moderate disease or severe disease, in the early stage of the disease or in the late stage of the disease. So according to that, again, the protocol will vary, because there are companies or there are institutions who have asked for early stage, and there are institutions who have asked for a later stage.
So now on the stem cell part, there is a question by one participant who is asking that using stem cell can develop artificial blood cells and organs.
Yes, they are trying. A lot of research is going on. There are companies which have come out with artificial platelets. There are companies which have come out with artificial hematopoietic stem cells, and then again, the organs have also been tissue engineered. Professor Anthony Atala of Wake Forest University has done a tissue engineered bladder, and he had implanted the bladder in patients who have got defective bladder from birth, and then there is a follow-up of more than 10 years. So there are a lot of work which is going on. Not only blood components, but organs have also been tissue engineered.
Another question is that the patients who have recovered from multiple viral infections, can their plasma also be utilized?
Actually, yes, the studies have come out that patients who are not exposed to COVID, they have neutralizing antibodies, adequate neutralizing antibodies, probably because of a cross sensitivity between the SARS and the other coronaviruses and the previous infection which the patient had. So he has got antibodies which will be able to neutralize the present COVID. So there is a possibility, already it has been proved that cross reactivity is there, but how much, again, as I said, how much antibody titer is needed, whether you have the adequate titer to ward off this particular viral infection, that has to be assessed.
So another question is, lyophilized plasma is suitable for any particular blood group?
Yes, actually it is blood group matched. It has to be matched. Either it has to be AB positive, or AB positive is universal donor for the plasma. So either it should be AB positive, or it should be blood B to B, or A to A, or O to O. So it should be matched.
Another question is that does the treatment to pulmonary infection, which is caused by bacteria like pneumonia, stem cell therapy has a role?
Yeah. There are actually the one trial which now they are doing by the company Athersis. They have got an approval to try for COVID, but actually their trial was for a pneumonia. It was for another indication of ARDS, not because of COVID, but other causes of ARDS. So they had already crossed phase 2. So the same product now they have applied for COVID. So already there are studies going on where bacterial pneumonitis and other causes of ARDS are being addressed by stem cells.
So let's take the last question, which will keep us in time. Is that is there a specific time to collect the stem cell, and from a person? Maybe this guy may be asking about the circadian rhythm, if they have any role in stem cell collection.
Yeah, circadian rhythm, definitely there is a role, but we can't take it in the middle of the night. Yeah, so actually probably I don't know, but this is a good question. I would also like to know which is the best time, because it is during sleep that we are able to regenerate most of our organs. So yeah. But the hospital working time is there, so probably it is only in the daytime when we can collect suppose a patient from the bone marrow or from the fat we want to collect, or even a dental pulp. It is only mostly in the daytime we do. But I don't know if nighttime is the best time, then we have to think over.
Sure. So, okay, so we have covered a lot of question, and some more question will be forwarded to you, sir, by Dr. Logan later on.
Sure, sure.
And we
Sure.
take this opportunity and thank you so much for your very insightful talk, and we would like to give you a thanks.
Thank you, sir. Thank you so much.
So formally thank on behalf of organizing committee, I would like to thank the speaker, Dr. V.R. Ravi, for giving an excellent talk about stem cells and convalescent plasma therapy and lifeless convalescent plasma. Especially, he explained about the Brahmastra weapon against COVID-19. That is lifeless convalescent plasma. That is very, very interesting, sir. Thank you very much. And also, he has explained that we are congratulating the Dr. Ravi and his team for their clinical research work on regenerative medicine to treat many diseases like bone fractures and cornea defect and so on. And thank you very much and addressing all these issues here, sir. Thank you very much. And also, I want to extend my sincere thanks to Dr. Sanjay Kumar for sharing 3 different sessions in spite of his busy schedule. Thank you very much, sir. I want to thank participants. Nearly around 618 participants participated in this particular session. Thank you all the participants. For the participants' information, the next session will start at 7 p.m. That is, will be a keynote speaker from Purdue University, Professor Philip S. Love. Officially, we are closing this session. The next session will start at 7 p.m. You can join using the same link. Thank you all. Thank you very much.
Thanks.
Thank you. Nice time.